Bismillahirrahmanirrahim. Assalamu alaikum. Myself Isha Tariq Bhatti, MPhil Pharmaceutics from Punjab University, and I'm working as a lecturer in Faculty of Pharmacy at Very University. So this is my eighth lecture of biopharmaceutics and pharmacokinetics, and the topic of today's discussion is protein binding and displacement. So we will see what are the learning outcomes of this lecture. First of all, we will study about protein binding, then displacement, then mechanism of drug displacement, then importance of displacement, then effect of displacement on distribution parameter, then distribution parameters, and kinetics of protein binding. So we will start our lecture with protein binding. So protein binding is the complexation of drug to plasma protein is known as protein binding. What happens in protein binding? A protein molecule that is plasma protein, it binds with the drug. So it will form a drug protein complex, which is known as protein binding complex. Protein binding occurs due to interaction of drug molecule with the binding sites available on the protein molecules. So the drug, it will bind to the protein molecule at the binding site, which is assumed to be preformed, and the molecule which form is known as drug protein complex. Then these binding sites are assumed to be preformed, but binding of one drug may also modify the configuration of the protein in such a way that the drug binding influence the nature of binding of further molecules of drug. So once the drug bind to the protein molecule on its binding site, it modify the configuration of the protein in such a way that it influence the binding of further molecules of drug with that protein. We can consider here the example of binding of hemoglobin to the oxygen molecule. Once the oxygen molecule bind to the hemoglobin molecule, it modify the configuration of the heme group or heme protein in such a way that it influence the further oxygen molecule to bind to it. So this is an example of protein binding. Now we discuss about drug displacement. What is displacement? First of all, we consider this, that what is displacement? When a drug that is a displacer compete for the same binding site on the protein already occupied by the drug, which is also or maybe called as objective drug, this is called as displacement. So when a highly protein-bound drug is displaced from the binding site by the second drug, a sharp increase in the free drug concentration in the plasma may occur. This leading to sudden increase in drug concentration or toxicity. What happened here? When a drug that is, as you know, that drug has some affinity for the protein, for the protein at the binding site, so that due to this affinity, for the due to this affinity for the binding site of the drug, drug can bind to the protein. So when a second drug came, and this second drug compete with already bound drug for the receptor side or the binding site, and the second drug which comes here is have the more affinity for the binding site. Then what happened? The drug that is already bound, it will displace, and the that is the second drug, it will take its place. And as a result, as the drug that is already bound, it becomes free. So the free concentration of this drug, that is the first drug in the plasma concentration, increases, and this sudden increase in concentration of the first drug will lead to sudden increase in drug concentration, which can lead to toxicity. As this concentration is very high, so it lead to toxicity. Then comes, for example, an increase in free warfarin level, increase in bleeding when warfarin is co-administered with phenylbutazone, which compete for the same protein binding site. As phenylbutazone, it has more affinity for the binding site. So what happened? If the warfarin is already bounded, so what happened? When the phenylbutazone is co-administered with warfarin, what happened? It will displace the warfarin from the binding site and take its place, and the level of warfarin in the blood will suddenly increase, and this will lead to the bleeding. And this is the example of toxicity reaction or the displacement reaction. In contrast, a drug that is extensively bound and actively secreted, such as penicillin, has a shorter elimination half-life because active secretion takes preference in removing the drug from the protein as the blood flow through the kidney. So what happened here? If the drug, say for example, it is penicillin or cephalosporins, what happened in the case of penicillin or cephalosporins? When they bound to the plasma protein, as they are actively secreted and highly protein bound, what happened? Um, when their concentration increases, the excretion parameter, that is active secretion method, it will take preference, and it will causes the drug to remove from the body. As the blood flow to the kidney, it will causes the drug to get eliminated from the body. Displacement is more critical for highly protein-bound drug than a weakly bound because high percentage of drug is displaced on administration of the second drug. This displacement phenomena is more important, or or you can say that this is more critical for those drugs that are highly bound to protein, that are highly bound to protein. As you know that for a weakly bounded drug, when it is displaced from its binding site, the concentration of drug that is available is not that much high as it is available, as in case of the drug that is highly bound to plasma protein. If the drug is, say for example, 95% bound to plasma protein and it is displaced from its binding site, then the free drugs that will be available into the blood will be very high. As compared to if a weak drug that is 35% bound to plasma protein, when it becomes freely available, its concentration that is available in the plasma concentration will not be that much high. So it is more critical phenomena. um, Here we consider an example. There are two drugs, that is drug A and drug B. Drug A is highly bound to protein, and drug B is weakly protein bound. So what happened? As you know, in normal case, the bound fraction of the highly bound drug is 95% and the free drug is 5%. And in case of weakly bound drug, 50% is bound fraction and 50% is free fraction. So a displacer displays 5% of bound fraction, um, and bound fraction of objective drug. So the bound fraction, that is 90% and the free drug is 10%. So this will increase the increase 100% of the free drug, and this will lead to 100% increase in the free drug concentration. And the bound drug, as in case of weakly bound drug, bound fraction is 45% and free drug is 55%, increase in 10% free drug. So it is more critical in case of highly protein bound drug. Then comes with drugs that are highly bound to the plasma protein, a small displacement from the protein causes a transient increase in free drug concentration, which may cause a transient increase in pharmacological activity. As the free drug concentration is, as the drug is displaced from its binding site, the free drug concentration will be suddenly very high. So the pharmacological activity, as the available free drug concentration is high, so the pharmacological effect will also be high. So this is the case which is explained in this slide. Then, however, more free drug is available for both renal excretion and hepatic biotransformation, which may be demonstrated by the transient decrease in elimination half-life. So as the more free drug is available for renal excretion and hepatic biotransformation, so this can be demonstrated by transient decrease in elimination half-life. So the elimination half-life will also be decreased. Then mechanism of drug displacement. Drug displacement from the protein by a second drug can occur by direct competition for the second drug for the similar binding site or alteration in protein configuration. The displacement of protein by the second molecule can occur. Okay, this displacement, that is a drug is displaced by the second drug molecule. So this displacement um, is due to direct competition, that is the both drugs are competing for the same binding site, and the drug that has more affinity for that binding site will takes the place of the drug that is already bound. So an alteration of the protein structure may also change the capacity of the protein to bound to bind drugs. So when the drug molecule, bind to the protein structure, what it will do? It will alter or modify the configuration of the protein. So it will also change the capacity of the protein molecule to bind to drugs that are to other drugs. Okay. Then, for example, aspirin acetylate the lysine residue of the albumin, which changes the binding capacity of this protein, uh, um, which changes the binding capacity of the protein for certain other anti-inflammatory drugs such as phenylbutazone. So as aspirin, it will acetylate the lysine residue of albumin. So it will change the binding capacity of this protein with other drugs which bind to the same protein. Say for example, in case of phenylbutazone, it binds to the same protein molecule. So when uh, as you know acetyl or aspirin is already bounded, it will change its capacity for the phenylbutazone, and the binding capacity of phenylbutazone will be decreased. Then comes importance of displacement. So this is very important phenomena, as it directly increases the free or unbound drug concentration as the result of reduced binding in blood. um, So it will increase the free or unbound fraction of the drug that is available into the blood, thus reducing the binding in the plasma protein. Okay. Increase the free drug concentration that reaches the receptor site directly, causing a more intense pharmacodynamic or toxic response. So when the free drug concentration, it will be increased, the receptor site, it will when the free drug concentration reaches to the receptor site, or increased drug concentration will reaches to the receptor site, it will causes the more intense pharmacodynamic effect, or it will lead to toxicity or toxic response, as the concentration of the drug that is available to the receptor is very high, so it lead to toxicity reactions. Increase the free drug concentration, causing a transient increase in volume of distribution and decreasing partly some of the increase in free plasma concentration. As the increase in free drug concentration, it causes the transient or sudden increase in volume of distribution. So this increase in volume of distribution, this will decrease partly some of the increased free plasma concentration. We can say that this is the mechanism which will reduce the effect of that high concentration that is suddenly available into the blood. Then increasing the free drug concentration, resulting in more drug diffusion into the tissue of eliminating organ, particularly in the liver and kidney, resulting in the transient increase in drug elimination. So increase in free drug concentration, it will result in more drug diffusion into the eliminating organs, and particularly in the liver and the kidney. So this result in transient increase in drug elimination. So it will, the free drug concentration is suddenly increased, what will it do? It will trigger the other mechanism, or you can say it will trigger the homeostasis system, so that the concentration of the drug can be maintained into the body. So what will it do? It will stimulate the eliminating organs to eliminate the drug, or you can say that drug elimination um, rate uh, is increased due to this effect. This is, you can say, a compensatory mechanism which will try to compensate the amount of drug that is very high drug concentration into the body. So it will try to reduce that concentration. So displacement produces the basis for drug interaction. So this reaction, as displacement is very critical reaction, as it suddenly increase the free drug concentration. So this free drug concentration can lead to different drug interactions. So this is the basis for drug interactions also. So now we will consider the um, effect of displacement on distribution parameters. So drug displacement lead to first of all increase apparent volume of distribution, then increased half-life. and clearance will remain unaffected. Then comes distribution parameters. These include apparent volume of distribution and distribution half-life. Half-life and volume of distribution are the important parameters in case of displacement. So now we consider the kinetics of protein binding. So there are two parameters which are of immense importance, that is binding or association constant and ratio of moles of drug bound to total moles of protein. And here is the equation that is described as K association, that is binding constant or association constant, is equal to PD divided by P plus D. Here P is the protein and D is the drug. So PD is the protein drug complex. So R is the R is so from this equation we can conclude that R is equal to NK association D divided by 1 plus K association D. So where N is the mole of proteins and K association is the association constant, P is the protein, and R in term, and if we explain R is the regression constant, R is the regression. So R in term of dissociation constant, that is regression in term of dissociation constant, will be equal to, or it will be given by NK dissociation into drug 1 plus K dissociation into drug. So this is the fraction for, or you can say expression for this equation. The above method is not straightforward method, as the kinetics of protein may be non-linear or saturable. If the kinetic is not straight line, then it, or you can say that it may be non-linear or saturable. So a graphical method has been proposed, which is known as double reciprocal method, in which we convert the curve graph into the linear one. From this graph between from the graph between 1 over d on x-axis and 1 over r on y-axis gives the value of 1 over n, and the slope is 1 over n k association. So in this graph, as we have reciprocate, or you can say reciprocate the both term d and r, so it will be given as 1 over d and 1 over r, 1 over d on x-axis and 1 over r on y-axis. So this method is effective only when the graph is straight. So you can see the graph here, that this is 1 over r and this is 1 over d, and this line is indicated as 1 over n, and the slope of this graph will be equal to 1 over n k association. So this is the graph for double reciprocal equation. And if we calculate, if we put the value of 1 over r, as the value of r is equal to n k association plus d, 1 plus k association d. So we reciprocate this equation as following. So if we, 1 over n, 1 over r is equal to 1 over n k association plus d plus 1 over n. So if we consider another method, that is scattered plot method, in the scattered plot method deal with the complex nonlinear processes, and from the graph between r on x-axis and r by d on y-axis, the y-intercept gives the nk association and slope is minus k association. So curved linear line shows involvement of more sides. So this is the graph. So this is r on x-axis and r over d on y-axis. Slope is minus k association, and the y-intercept is equal to n ka, and the x-intercept is n. So this is the graph that shows scattered plot method. So if we consider in vivo method, it will be described as db over df is equal to nk association pt minus k association df. So where dB is the concentration of bound drug and dF is the concentration of free drug, and PT is the total protein concentration. Um, so um, the summary of this lecture is that we have considered protein binding, then displacement, then we have discussed mechanism of drug displacement, then importance of displacement, then effect of displacement on distribution parameters, and then distribution parameters, and then kinetics of protein binding. So thank you so much for listening. So if you have any question pertaining to this lecture, you can ask me on Google Classroom discussion board. So and the class code for Google Classroom is mentioned as it is mentioned here. So thank you so much, students, for listening.